Hello, and welcome back to the Computational Genomics Lab. We have finished all of our analyses of our RNAseq DE-seq2 results, and now it's time to make some figures from them. And my favorite way of making figures is ggplot. We're going to do a few classes on ggplot, get a feel for how it's set up and how we run it. But to get things oriented, we're here on the GitHub on Masterclass, Lessons, 07 already, visualization and figures, and we're going to be doing 01 plotting in ggplot. Okay, so let's get started, and I'm going to go here and make sure I clean up all of my environment. This is a good lesson to learn, not to leave all your stuff hanging out overnight. Okay, so we were previously in 06, so we're going to go to 07 here, visualizing ggplot 1, so we'll have 3 short little classes on ggplot because there's a lot to learn and it's so powerful. You can make the most beautiful figures with ggplot, and I would say one of the best advantages of R overall is plotting with ggplot. Okay, so let's clean out our environment too. Okay, fresh start. So what I have written down here is a lot on ggplot background and some resources. We're going to need to load ggplot. You've installed it by now, there's layers in ggplot. So you put in your data, your axes, and then you can add more and more aesthetics and geometries and all kinds of fun things. And so these are the 8 scales here, shown here. And we're going to get started with just the basics. And again, our markdown files on the GitHub are going to have a lot more information. I'm going to go fairly quick through the videos on this. So we just finished with our TPM results, and we had all those, the average and standard deviation TPM per time point. And so let's load that in. Okay. And the next one is going to be our DE-seq2 results, which was that ResDF and filtered ResDF that had our p-value and log fold change cutoff. Okay, so layers 1 and 2 are the mapping layers and the data itself. That's going to be plotted. And you can look into ggplot. I strongly recommend it. There's a lot on Google, a lot of good resources here. But we're going to start with just a simple example. We're going to take our filtered ResDF. Now again, that's the DE-seq2 results compiled for all time point comparisons, filtered to p-adjusted of less than 0.05, and a log fold change of either 2 or less or half down. So it has to be bigger than that. And so we can just go into that with ggplot, similar like we did with tidyverse, and pipe the data to the next command. And so the first thing ggplot wants is what is my data at? Okay, and this AES is the axis layer or the aesthetics layer. And anything inside there, and you can do a ton. You can do functions, all kinds of stuff we'll get to in a bit. But for now, we're just going to say the x-axis equals the log2 fold change column, and y equals the p-adjusted column. Now, these are not unique. We did that last time where we found the number of unique genes, but that's okay because some genes can change in more than one time point, which we have there. And so we basically told it to go into this data frame, get this p-adjusted value, and plot it. So it knows all the information inside that object, and we're going to make it then with a plus is where you go to the next layer. So we're going to say do this and then add this next layer. And that's kind of like the pipe in Bash and in tidyverse. And we're saying make it a geom point. And again, on the web, you can find tons of your favorite ways of plotting things. And so we run that. Look at that. Already we have a plot that's very satisfying. So here what we can see on the x-axis is how much the gene changed, and at least twofold, and the p-value that corresponded to. So we can see some out here that are 2 to the negative 20th and 2 to the 20th. And most of our data is down here in the middle. So we're already starting to find out results here. That our p-values are really centered around smaller fold changes, but they're still significant and reasonable. Okay, so let's say we want to change those axes. Instead of minus 20 to 20, we want to change it to 10 to 10. So we have the exact same code here, put in my data frame, tell me what the axes are with this AES function inside there. X equals log fold change, Y is P adjusted, G on point. Now we're going to add another plus to add xlim, which is the x-axis limits, and there's a y-lim as well. So you can modify your plot to get a little more zoomed in. If we do that, we can see it looks very similar, and we've just cut out those few points that were way out there. And we can zoom in even further by saying xlim equals -5 to 5. You'll notice in hist, when we did the hist function, you had to have the C and then the parentheses with the two axes separated by a comma. In this case, you don't need the C. Okay, so just zooming in slightly further here, and we can see a bulk of our data is in this range. And we have no data in here because that is what we filtered on. Okay, so we can change the colors. So this is in the G on point layer here where we're telling it to make a dot plot essentially. And we can, there's all kinds of fun stuff. Again, there's way too much to do in one class, but Google what fun plots can you make in ggplot, and you'll know the logic now. So we're going to go use that same data frame, same axes, G on point, but instead of leaving it blank like we did up here, we're going to put in some parameters like shape equals square, color equals purple. There's also color codes you can use, and we'll use this smaller xlim. Here's a web link to a bunch of good shapes and colors for ggplot. And so cool, look at that. So now our plot got some sort of flair to it, and we can see they're squares instead of circles, and they're purple. Okay, one of my favorite colors. So let's now do something pretty cool where now instead of in the G on point, we're going to leave it blank. So we're just going to get that same black circles as our data. But look at what we can do in this AES layer when we're mapping the data frame to the plot. Okay, so we have the same things here. X and Y are log fold change, P adjusted. And now we're going to color it by result name. If we go in here, we see that there are multiple different results names like 12 versus 0, 48, 96, 24. So we are going to get our colors of our dots all on the same plot, but we'll know which time point it came from. That sounds really handy. Okay, cool. So look at all the information we just got in 2 lines of code here. So we can see now that purple is our last time point, and they seem to be down here on the bottom. So maybe at the later time points not much is changing, but let's see, green and blue seem to be a majority of the dots here. So we're already finding out a result that it looks like most of the changes are occurring to our genes in the 24 to 48 hour time points. Cool. Okay, so this is really the same thing, except for now we're going to specify just one of those result names, and it's written a little different. For clarity here, we have our data frame, x-axis, y-axis. It's all the same. It just has an enter in it. And the color equals result name. We did that last time, and it gave us the 4 different colors. However, now we can set it directly equal to the 96 versus 0 and see just how many genes had changed only in that time frame. Okay, so we see there's still a lot of genes changing. And, oh, actually not. So false means it was not the 96 time point, and that's most of the data. And blue means it's true, which is a very little of the data, and it's all kind of in here. But there are some of those big changes out here that occurred. So we're already starting to dig into the data and get results and find out when our genes were changing due to the DOX exposure in mouse stem cells. Okay, even better, we can use this function facet. And again, I'm going to explain all this in text in the R Markdown, so I'm going to go just kind of run through these. Same setup here, x-axis, y-axis, and now we're adding a plus with facet wrap. Now that's a function in ggplot, and what's going to do is make a figure for each of the time points. And we're going to say result name tilde dot, which means facet by result name, and you could facet by multiple things with that tilde having another object, and we'll get to that in a minute. But that's just kind of the nomenclature, at least I use. There are other ways to do this, but if you want to make all the plots of any unique value in a row, this will do it automatically. Pretty cool in my opinion. Okay, so if we run that, wow, cool. So we got 4 different plots here, and we can see how many genes change in 12 versus 0, 24, 48, and 96 hours, all right next to each other. And now we can really tell that a majority of our genes are changing in the 24 to 48 hour range. And once they've changed, they're probably not changing anymore at 96, and 12 hours might have just been a little too early for the docs to be doing its thing. Cool, great results. I've also started in the Markdown putting down results I think are interesting, so we could think about trying to write a paper down the road and just archive all of our results as we're learning ggplot. Okay, so here we are doing a histogram. So there we're only going to really have an x-axis, and the y-axis is by default going to be the frequency of those events. And so we have our ggplot function grabbing this data frame. The AES is now just x equals log fold change. And we're going to see how often genes are changing in each bin of fold change. And then we use geom histogram for that. And I have the question mark thing here to run. Tons of parameters again that you can add and make it more fancy, but this will do the trick, sort of. It's not very pretty, but we can see a vast majority of our data is in this log fold 2 range. Okay, log fold 2 of 1, or -1. Okay, so we can make more bins and get some more granularity in here. So now, instead of leaving geom histogram blank, we're adding bins equals 300, and we're changing the limits to 5 and -5. You can see how this kind of is building up, and you would do a lot of the same things over and over again. And cool, look at that, that looks much better. Okay, so now we can see that there are some genes out here, but not as many, and in each bin that we gave it 300 in this case, it's going to give you how many had a value in that bin. And so we can see kind of that maybe a few more are going up than going down, but it's pretty evenly split here. Okay, so now we're going to do the same thing, but we're going to fill in the AS layer here. So we have our log fold change as the x-axis, and we're going to do fill equals results names. And so you can see you can do a lot in this AES layer, and we're going to do even more coming up. Okay, so we have the geom histogram is the bins, same as last time. We're just adding this fill equals result name. And so cool. So now we not only have the same data here, but now we can see which color is which, and you can make use a dodge function in here to have them not overlap, but we can see pretty clearly again that 24 and 48 is the green and blue, and it's most of the data. So we already have a good result that things are changing most at those time points. Okay, so we're going to do the dodge. All right, cool. So that was a little hard to read because they're all overlaying each other. But so we're going to do fill. Again, we need fill in the data layer because that's actually in the data frame. So the AES layer has to go in and go into result name and say, oh, there's 4 unique values here. I'll color them by each of those 4 values. So all the same, bins, histogram, we're just adding now this position function to equal dodge so they don't overlap. And we're going to shrink the xlim a little bit even more to -2 to 2. Okay, let's zoom in here. And so now you can see with a little more granularity that most of this data is in the blue-green region with some of the reds and stuff down here in the bottom. Okay, same result, different ways to plot it. Okay, density plot is another great way. It's kind of like a histogram, but a little simpler to read. So we can do same thing, ggplot, same data frame, x-axis is log fold change, fill by result name, so we'll get those 4 values on the axis, and do geom density is now the new plotting layer we're adding instead of histogram. Okay, so let's take a look at that. Cool, so we see that you can see the sort of shape of the line now, and that 96 is changing mostly in here. It's going to fill it all in, so we don't actually have data from -1 to 1, but it's going to make a continuous plot. And we see a lot of the early time points here are very small changes, and then at 48 hours we have a lot more going up. Okay, so that's interesting. That's another result we could take note of. Okay, so then let's do this and change the alpha in the geom density. So everything is the same here. We're just changing the alpha, which is the opacity, essentially, of the plot. Okay, so let's take a look at what we get here. And now we can see a little better, some muted tones here, but now we can see each of the distributions overlapping each other. There. Cool. Now, what we're going to do is we're going to say which genes had a p-adjusted. So we're in this AES layer. Again, I'm just typing a little different so you can see that these are all the same things. It's just in a different structure. So we take our data frame. We set up our AS, x equals log fold change, as we've been doing. Fill, now you can do a function. We're essentially saying go into the p-adjusted column and only fill them if they're less than 0.01. Our cutoff was 0.05, so we're going to find the ones that are sort of more significant. We're going to color by result name, and we're going to do a density plot with an alpha of 0.3 and change the x and y axes. Okay, let's take a look at that. Cool. So now we can see that the true means it was p less than 0.01. Seem to be way out here, and we have a lot of these high density here on the log fold 2 of 1, which is twofold, but they're not p-adjusted less than 0.01. So lots of information in one plot here. You can also export these to a PDF, and you'll have them in vector, and you can change the colors or put them into Illustrator or something to that effect. Okay, so let's do one more here with the same thing. We're going to do fill equals result name, color equals result name. So we're just adding both the things we just did. And a geom density plot with an alpha of 0 and an x and y limb. So only the line will be colored if the alpha is equal to 0. There's 100% opaque. So that's a little easier to read here. So now we can see the 96 time points are changing quite largely. They have big changes. And again, most of the data is early time points are going up, so that's interesting. This is asymmetric, that the earliest time points, the genes that are changing are going up, and then only at later time points are they going down. Great. Again, lots more detail in our markdown, and we are going to continue our journey. This is just really the very basics of what you can do with ggplot, and you could probably study ggplot for the rest of your informatics life and figure out different ways of doing things. But we're going to get deeper into the other layers in the next class, and for now we have some results, and you can go through our markdown, and we'll see you then.